Hello, and welcome to our tutorial on how to use the AnnoJ browser to view neuron type-specific DNA methylation profiles as generated using whole genome bisulfite sequencing. So when you come to the browser, you're probably interested in a particular gene. Let's say, for example, you're interested in the inhibitory marker GAD1. We can come to the text box here and type GAD1, and then any genes matching that string will show up in the selection menu here, including multiple isoforms. So we can just select the only option in this case. And the browser now is going to jump to that position in the genome. Alternatively, if we knew the chromosome and position of our gene, we could have directly typed it in here in the chromosome and position section and clicked Go in the menu. In the gene annotation section, we now see our gene listed. The gray line to the right of the gene name indicates the gene body, whereas the green rectangles indicate the exons. At the current zoom level, we can't fully see our gene, so we can come over to the red magnifying glass and zoom out the browser. Now we can finally see the full gene body for GAD1. Just as a final note, the arrow at the far right of the gene is indicating that GAD1 is on the forward strand. So now let's examine the DNA methylation profiles that are shown in the main area of the browser. If we look at the far left of each track, we see that there's a label for each one. For example, the first one is showing CG methylation for mouse layer 2-3 cells, the second one is showing CG methylation for mouse layer 4, and so forth. Now if we scroll down in the browser, we see that there's first the CG methylation for excitatory cell types, then we have CG methylation for inhibitory cell types. Next we have non-CG methylation for excitatory cell types, then non-CG methylation for inhibitory cell types, and then finally we have our bulk reference samples that were used in the study as well. You'll notice that the non-CG methylation is indicated by the blue ticks, whereas the CG methylation is indicated by green ticks. So how do we read the methylation tracks? Well, in the center of each track is a dotted black line. The ticks above the track indicate methylated cytosines on the forward strand, whereas the ticks below the track indicate methylated cytosines on the reverse strand. And the height of the tick corresponds to the percentage of cytosines that are methylated at that position. So you can see some ticks go all the way to the top, whereas some ticks have a much lower height. One important feature of the AnnoJ browser is that you can actually zoom in to the base level position to view DNA methylation at individual cytosines. And so I've loaded up a separate browser view that is all the way zoomed in at the individual cytosine level, as shown here. From this view, we can see that there are two sets of CG sites that are showing some degree of methylation in at least some of the samples. The bar on the left corresponds to the cytosine on the forward strand, and the bar on the right indicates the cytosine on the reverse strand. And as I mentioned just a moment ago, the height of the bars are indicating the percentage of cytosines that were identified as methylated at that position. As you can see, the CG site is methylated in some of the cell types, such as the inhibitory cell types particularly. However, numerous excitatory cell types are not showing methylation at this CG position. So now let's return back to the main browser view that is showing a zoomed out version of GAD1. The blue bars beneath some of the tracks indicate hypomethylation in particular cell types at statistically significantly differentially methylated regions. For example, there are large numbers of blue bars underneath the inhibitory cell types throughout the gene body of GAD1. This indicates that there's significant hypomethylation in inhibitory cell types throughout GAD1, which is what we would expect because it is a marker of inhibitory neurons. There are a few final features that I would like to point out about the browser. First, you may only want to view some of the tracks and be able to delete some of the others. So the way that you can do this is either by clicking on the red minus sign here, or you can find the track over here that corresponds and double click on it. This will remove the track from the browser. Now if you want to add tracks back, you can come down and scroll down to here, go into the DNA methylation section, and you will see those tracks listed here. We can double click on them, and then it will add them back into the active window, and then you can drag the track back up to its former position as such. and now it shows back up in the browser where it was before. Thank you for viewing our tutorial. If you have any additional questions, you can find more information at our website.